All right, the composition of blood. It's a fairly complicated fluid. You have about 5 liters of it in your body. Somewhere between 45 and 60% of that fluid is plasma, and somewhere between 40 and 55% of that fluid is what we call formed elements, which is a really awkward way to say cells or parts of cells. And we'll see why they chose that awkward term. And really, for you guys, what you'll end up actually using from that number is there's a term called hematocrit, and this would be a lab value that you would get from a blood test. And hematocrit is the percentage of blood that is made up of those formed elements. So red blood cells, white blood cells, and platelets. And that's going to be an important lab value. Let's start by really looking at the plasma. I happen to be a big fan of this picture, but this is the one from your textbook and it does lay it out very nicely. It is very obvious when you look at the plasma that it's mostly water, right? Mostly water. But carried in there, we have proteins and solutes. So the proteins are a really important part of this plasma. The most common protein group, they're called albumins. This is going to be super important as far as maintaining osmotic pressure. So these albumins are one of the major things that draws water into the bloodstream. So if you say had liver damage like hepatitis and the liver stopped producing the albumins, that's where they're made, as those levels decreased in your bloodstream, you would start to see lots of edema, lots of swelling, because there's nothing pulling the water into the bloodstream. Those albumins can also be used as transporters for like our binding proteins for carrying hormones, or also maybe for transporting fatty acids. The next group of proteins are the globulins. Included in this group would be things like antibodies. So the other way to say antibody is immunoglobulin. So it's a globulin protein. Also the lipoproteins. So if you've heard of HDLs or LDLs, those would be globulins. And again, these can also act as hormone binding proteins. The final group of proteins, a much smaller amount, is this this protein called fibrinogen. And anytime I see this ending, OGEN, that's going to signify a protein that's like not in its final useful form. So what we'll see when we get to clotting is fibrinogen is converted into fibrin, and that's actually the protein that helps us form a really strong clot. So it's kind of like a precursor. So OGEN, watch out for that ending. That could be helpful. All right, so that's the plasma. We still need solutes. So solutes. So we have a whole list of stuff in here as well. So ions, right? Basically, I tend to say this as electrolytes, right? Remember, an electrolyte is just a salt that dissolves in water and is capable of carrying an electrical charge. And so you can say it either way, ions or electrolytes. Those are going to be important in here. We've got lots of nutrients, right? So think about all the different organic molecules, things like your glucose, right? Any of the carbohydrates really, right? But glucose is the common one. Amino acids, lipids, those are going to be all being carried here in the plasma. Waste products, things like urea, bilirubin, absolutely. And then also some of our CO2 and O2. There's very little dissolved actually in the plasma. We'll see that the red blood cells carry most of that. And then again, regulatory substances, they're talking about things like hormones and enzymes. Okay, now if we go down to the formed elements, and again, the percentage of blood that's formed elements, that's your hematocrit. Should be somewhere between 40-55%. Males average around 46%, females about 42%. So 45 that they're giving you here is actually a really nice number. Males have a slightly higher hematocrit than females, generally due to androgens. So testosterone or any of the androgens do help stimulate red blood cell production. Okay, so in the formed elements, we really have red blood cells, whose technical name is an erythrocyte. We've got our white blood cells, which are the leukocytes. I'm going to go back to my picture because I think it gives you those terms. So white blood cells, the leukocytes, those of course are important in the body defenses. The red blood cells, primarily it's carrying oxygen. And then platelets, this is what makes formed elements tricky. They're not really cells. Platelets are actually fragments of a cell called a megakaryocyte, and those are going to be important in clotting. There's that same list coming from your textbook, table 19.2, if you're looking for that. All right, let's look at these formed elements in a little more detail. So again, the red blood cells, really their role is carrying oxygen and CO2. In a single drop, you don't have to memorize these numbers, but in a single drop of blood, you've got 260 million red blood cells, right? 1,000 times more red blood cells than white blood cells. If you've looked at blood microscopically, which it's going to wait till next week, but we will be looking at them, way more red blood cells than white blood cells. Now, red blood cells are shaped like a biconcave disc, which could be excellent knowledge for trivia night, but you might also be going like, who cares? Basically, this shape where it's kind of like, it's a disc, but it's concave on both sides. It's kind of hollowed out. It gets this shape because it actually loses its nucleus during development, right? It gives the nucleus the boot so that it can carry that much more hemoglobin and therefore that much more oxygen. This shape also gives it a really large surface area per its volume, right? So huge surface area for the volume of the cell means that we're going to get really fast gas exchange, which is what we need when we pass through the lungs or working tissues. Okay. Another nice thing that happens with this shape is that the red blood cells will actually stack up like dinner plates, like you see here in this picture, and that helps give them like this smooth, they call it laminar flow. It's a nice smooth flow, so it's not all turbulent, and so the shape helps with that. The other thing that is so crazy to me is red blood cells, when they're going through capillaries are often actually smaller than the red blood cell themselves. They can be half the size of a red blood cell. And so this shape actually allows it to bend and flex to fit through there. So think about, like the little flexy dog frisbees you can actually like fold, or I don't know, they make tents or hats or something like that too, right? So it's the same idea. This cell can fold and then squish through really small blood vessels. So these red blood cells, they last about 120 days. They're traveling that whole time, right? It takes about a minute for a red blood cell to get through the whole body. And so they're going, in their 120-day lifespan, they're going like 700 miles, and then these guys are getting worn out. So they're not a particularly long-lived cell. And so what that's going to mean is that in your body, in your bone marrow, you're making something like 3 billion new red blood cells every second, right? A third of the cells in your body are actually red blood cells. And so what this image is trying to show you is how red blood cells are formed. And so I'm going to zoom in here a little bit. This is an image from a different text, but it gets to the same idea. So one thing that is key, flipping back, is that when blood cells are forming, where's my pen? You have a stem cell, right? And stem cells are just going to undergo mitosis and keep replacing themselves. But really early on, we end up with 2 different groups of stem cells. Myeloid stem cells basically make all of our cells except lymphocytes, and lymphocytes come from this lymphoid stem cell. So red blood cells are starting out from this myeloid stem cell lineage. Right? Now, there's a lot of different steps in the development, but I think there's a few that you should actually recognize. And so I've circled them here. You should know that it comes from this myeloid stem cell lineage. Early in its development, so they're saying day 2, notice this term erythro. That should make you think red blood cell. Anytime you see, so erythrocyte, erythroblast. Oh, and if you think back to bones, we use that terminology before blast is typically indicating an immature cell. So here we go, erythroblasts. We're going to spend a few days, this is going on in the bone marrow, red bone marrow. And then right before the cell enters circulation, it's actually going to ditch its nucleus, right? So I said it kicks the nucleus out. And at that point, it becomes what's called a reticulocyte, right? That reticulocyte is actually going to be the cell entering circulation. So they do enter circulation before they're fully mature. And then you have this mature red blood cell. The reason I point this out is reticulocytes should really be like less than 1% of those red blood cells. But you could actually see these in lab values. And so if you saw someone with higher levels of reticulocytes, it should make you pause and go, okay, why is this person working to make so many red blood cells, right? I mean, if they just lost a lot of blood in an accident, that would make sense. But you might think about some other reasons that this would be going on. Okay. And then those mature red blood cells have about 120 days out in the bloodstream. For this process to work properly, right, and we said what, 3 billion new red blood cells a second, you need a lot of resources to make this happen. You need, you need nutrients like iron and amino acids to build the hemoglobin molecules. It takes a lot of B12 to make this happen. We're also going to choose, well, here's our hemoglobin, so there's the iron. Sorry, I'm just like, yeah, of course you need iron. In each hemoglobin molecule, you need 4 iron molecules. It's the iron that actually grabs onto the oxygen and gives hemoglobin this super cool property that I won't spend a lot of time on right now, but this is referred to as the oxygen hemoglobin dissociation curve. And the cool thing with hemoglobin is that this is not a straight line. Right? And we'll talk about this more with the respiratory system, but hemoglobin is really cool in that it will pick up oxygen, grab onto it, but then when it gets to the tissues that need it, it will let it go. And so we'll talk about that reversible binding much more. But we go through this process, of building more red blood cells, really in response to hypoxia at the kidney. The kidney has a really rich blood supply and is usually the first organ to notice a lack of oxygen. It actually produces a hormone called erythropoietin, EPO. So here's that secondary endocrine function of the kidney. And that's going to then go ahead and increase the erythroblast division, accelerate hemoglobin synthesis, all that. That is why athletes like Lance Armstrong would take EPO, trying to get more hemoglobin in their red blood cells, therefore in their blood, so that they can carry more oxygen. And like the crazy thing here too is we talked, like thousands of red blood cells, millions of red blood cells. Each red blood cell has 280 million hemoglobin molecules inside. Again, don't memorize it. But if each of those is carrying 4 oxygens, each red blood cell can carry 1 billion molecules of oxygen. So that's a lot. If you don't have enough hemoglobin, you develop things like anemia. That can be from low iron, low B12, any of those nutrients we were talking about. Okay, so 120 days and then a red blood cell is done for. At the end of that red blood cell's life, we don't want to lose all those really important nutrients. So ideally, the cells don't just rupture in the bloodstream, but instead, we get these old damaged red blood cells actually being engulfed by macrophages. So here's our old and damaged red blood cells, and this big purple grimace here is a macrophage, right? Particularly macrophages, right? They're white blood cells, part of that lymphatic system, so they'll be embedded in things like the liver, the spleen, bone marrow, and those macrophages are sitting there and kind of checking for these old red blood cells. If they get engulfed by a macrophage, the macrophage can help redistribute those nutrients, right? If they break in circulation, that might just get lost. So what we see happening here, right, is we take that heme group, that iron-containing group, and the iron just goes right back to the bone marrow. So there's a special transport protein, and we carry that iron back to the bone marrow. The other part of our hemoglobin then is going to have to get broken down more. So the protein portion of this is going to go through a couple steps here. So biliverdin, if you've heard of that, bilirubin is usually the one people recognize. Bilirubin is actually what's going to then enter circulation, go to the liver to actually get processed into bile, and then leave the body that way. If you have high levels of bilirubin in the bloodstream, it ends up showing up as jaundice, that kind of yellowish coloration. So that could indicate liver problems if the liver isn't actually taking that bilirubin in and converting it into bile. And I just think this is so wild, because when we get to the digestive system, we're going to talk about how important bile is. And here we are, our body is figuring out like a secondary use for this waste product, right? Waste not, want not. So then what's excreted in bile ends up in the intestines. And so this is going to be what ends up giving feces kind of the yellowish or brownish coloration. And then we'll also see some of that ending up back in circulation, right? So if you think about the intestines doing all this absorption, some of that waste product will actually end up in circulation, and then the kidney takes it out, and that's why urine has that yellow color as well. It's all this breakdown product from red blood cells. So this ends up being a really efficient process, particularly, I mean, the iron. To make all the red blood cells you're making in a day, you need something like 26 milligrams, right? But actually in your diet, you only need 1 to 2 milligrams because we're doing such an efficient job of recycling. And this is the same image of that from your textbook, figure 19.7. Okay, so now we have to look at the white blood cells, the lymphocytes. So white blood cells, and again, we kind of have this whole lineage coming down here, but when you zoom in and just look at these white blood cells, right, they are circulating briefly, right? They really don't spend a lot of time in the bloodstream. They're then going to actually move out of the bloodstream and migrate through the connective tissues of the body. So some of the unique things that these do, they migrate out of the blood. They can actually squeeze themselves through the endothelium of capillaries. They use amoeboid movement. So they're not just getting carried by the bloodstream. Often they're getting carried by the bloodstream, but they can choose following chemical signals, really, to use this like amoeboid motion, right, sticking out part of their body and kind of following it. They do use chemotaxis to decide when to do that, so they're following chemical stimuli. The famous example, right, is the site of an infection. A macrophage in circulation is called a monocyte. So this monocyte's floating along and catches a whiff of salmonella, some bacteria. He actually moves to the edge of the capillary, starts crawling, right, with this amoeboid motion along the capillary until he can find a place to squeeze out and now be at the site of infection. Macrophages and then neutrophils are phagocytic cells. I guess eosinophils count as that as well, but monocytes become macrophages as soon as they leave circulation. They can actually become a few different cells. And neutrophils, they're the ones doing phagocytosis. Eosinophils, they tend to handle more like large parasites. So it's not quite the same as engulfing them. But okay. So one of the things we'll be doing in lab next week is working on being able to identify these different white blood cells, thinking through their functions. And this is still an important technique, but I will say, if you go to the hospital and they want to know how many of each of those categories of white blood cells you have, because again, that's a very predictable. When they do a white blood cell count, typically it's actually going to be a computer. So they have a machine where light shines through the blood, and this is crazy, right? This is microscopic. It's basically going to pick up the pattern of the nucleus, right? So how complex the nucleus is, right? Increasing complexity this way, and how big the cell is. So notice, right? Big cells without a complex nucleus describes a monocyte. Let's take a peek. Oh, look, this monocyte is like the size of 3 red blood cells, at least. He's huge, and his nucleus is this big blob, so very not complex. Boom. So he falls out up in this range. Lymphocytes, how would we read this? Well, they're smaller, but they also have a not complex nucleus. Boom, lymphocytes. Look at that. He's tiny, right? Just a little bigger than a red blood cell. And again, just a circular nucleus. So anyway, the computer can make this much faster readout rather than a lab technician having to sit there and count all of these different types of white blood cells. All right. What else to tell you about white blood cells? Let's do this. Okay. So neutrophils end up by far being the most common white blood cell. Somewhere between 50 to 70% of all your white blood cells are neutrophils. They have this very segmented nucleus. You'll look for that in lab. They are phagocytic cells. Typically, they're using things like hydrogen peroxide or superoxide or enzymes to kill the bacteria or microbes that they've engulfed. They typically die after engulfing 1 to 2 dozen bacteria. So in this process of killing the bacteria, they're going to damage themselves. Eosinophils, I mentioned, are more important in things like parasites. They're very rare. They're actually only like 2 to 4% of the white blood cells. And they stain this really bright orange. So they have granules inside of the cell. You can actually see granules in these 3 cells, the little dots. These stain kind of this reddish orange, and they have this pretty obvious, they call it a bilobed nucleus, right? So it's got 2 major lobes. So they can be phagocytic, but they're usually using enzymes and nitric oxide to kill, and usually that's through exocytosis because they're going after big things, multicellular parasites. So you would expect to see higher levels of eosinophils in someone who has a parasitic infection or potentially allergies because they'll kind of treat allergens, which are relatively large, the same way. Basophils, extremely rare, less than 1%. They're small, they stain really darkly. And these are cells, they contain histamine. Histamine is important in the inflammatory response, help dilating blood vessels. They also contain heparin that helps prevent blood clots. Monocytes, somewhere between 2 and 8% of all the white blood cells. They're huge, right? They are really big. They have kind of this characteristic, they say a horseshoe-shaped nucleus. This one looks kind of more like a candy cane to me. These guys end up only staying in the bloodstream usually for like 24 hours, and then they're actually moving out into the tissues. Lymphocytes, 20 to 30% of the white blood cells. This is going to include your T cells, right? T lymphocytes. So if you think about helper T cells, they're helping coordinate the whole response. There's also cytotoxic T cells. They're going to go ahead and attack cells that are actually infected with something like a virus. Other lymphocytes would be the B cells or B lymphocytes. They're the ones producing antibodies. These guys are fairly characteristic. They're small and they have just that like thin layer of cytoplasm. That's usually what I look for if I'm trying to identify them. If you think about their production, again, this is pretty much taking place in the red bone marrow. They do a lot of their development in the lymphatic tissues, places like the thymus, the spleen, lymph nodes. So they've migrated there. They kind of take up residence. The production of these, right, we saw red blood cells being regulated by the hormone EPO. A lot of the stimulus here on white blood cells is actually exposure to antigens. We're almost there. Our third category of the formed elements here are the platelets. Again, these are cell fragments, which is why we call it formed elements instead of cells. And they're fragments of this huge cell called a mega karyocyte. And these platelets we will talk about when we get to blood clotting. Let's do that. Okay, wrapping it there.